Dear students, welcome to the MCQ discussions based on the chapter Biomolecules. Okay. First question, which of the following statements is or are true with respect to the composition of amino acids? Amino acids are substituted ethanes. That is a wrong statement. Amino acids are substituted methanes. So here carbon, hydrogen, 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 hydrogen. This is the organization of methane. Isn't it? So in the methane, one hydrogen is replaced by NH2 group, substituted by NH2 group. Another hydrogen is substituted by carboxyl group. Another hydrogen is substituted by a variable side chain R. Okay. So it's a substituted methane is called amino acid. It's not substituted ethane. So choice 1 is false. So when 1 is false, A, B and D will be. So C will be the answer. R group is the variable part of amino acid. So this is the variable part of amino acid. Amino group and carboxyl group are attached to the same carbon. You see, amino group is attached to this carbon. Carboxyl group is also attached to this carbon. This carbon is called alpha carbon. Okay, so 2 and 3 are right with respect to the composition of an amino acid. Next one is a simple question. Number of carbon atom in palmitic acid. Palmitic acid is the most abundant fatty acid in human body. The formula of palmitic acid, CH3, CH2, 14 times COH. So the number of carbon atom will be 1 plus 14, that is 15 plus 1, 16. So it's a C16. C16 type of fatty acid is called palmitic acid. Next question. Trihydroxypropane. Trihydroxypropane means CH2OH, CHOH, CH2OH. The 3 carbon propane. Trihydroxy, 3 hydroxyl groups are there. This is actually glycerol. Glycerol is nothing but trihydroxypropane. Next question, consider the following statement. Statement 1, unsaturated fatty acids possess one or more carbon-carbon double bonds. The unsaturation is due to double bond. That statement is correct. The common phospholipid found in cell membrane is called uridine. Uridine is not a phospholipid. Uridine is a nucleoside. Ribonucleoside. The phospholipid found in cell membrane is called lecithin. It's called lecithin. So here the statement 2 is wrong. So 1 is right, 2 is wrong. Answer is B. Next one, nitrogenous base that is totally absent in RNA. You see that nitrogenous bases are adenine, guanine, cytosine. These are present in RNA and DNA. But uracil is present only in RNA and thymine is present only in DNA. So RNA has 4 types of nitrogenous bases: adenine, guanine, cytosine, uracil. DNA also has 4 types of nitrogenous bases: adenine, guanine, cytosine and thymine. So nitrogenous base that is totally absent in RNA is called thymine. So thymine will be the answer. Next one is a tricky question. Match the following: adenine, adenosine, adenylate acid. Adenine is a nitrogenous base. Adenine is a nitrogenous base. Adenine plus sugar, adenine plus ribo sugar forms adenosine. Adenosine is a nucleoside. This is a nitrogenous base. Nitrogenous base plus sugar form nucleoside. Adenosine is a nucleoside. Then adenylic acid, phosphoric acid. When phosphoric acid is added, it is called a nucleotide or nucleotide. So this is adenosine, this is adenine, The combination is called adenosine. This combination is called adenyllic acid. Phosphoric acid is added. So it is called nucleotide. Adenine is a nitrogenous base, 1B. 1B. Adenosine is a nucleoside, 2C. So answer is C, 3A. Next one. Find out the number of pyrimidine bases in the following group. Adenosine. Adenosine means adenine plus sugar. Guanosine. Guanine plus sugar. Cytidine. Cytosine plus sugar. Uridine. Uracil plus sugar. Thymidine. Thymine plus sugar. That means all these are nucleosides. Find out the pyrimidine bases. They are not nitrogenous bases. All the given words are referring to nitrogenous nucleosides, not nitrogenous bases. What is a pyrimidine base? Pyrimidine bases are cytosine, uracil, and thymine. These are the pyrimidine bases. So the answer will be none. Next question regarding glycosidic bond. Glycosidic bonds are seen in between adjacent fatty acids in a fat molecule. In fat, ester bonds are there. Ester bonds are there. Nucleotides in a nucleic acid chain. In adjacent nucleotides are connected together by phosphodiester linkages. Sugars in a polysaccharide chain, that bonds are called glycosidic bond. Amino acid in a polypeptide chain, that bonds are called peptide bonds. So glycosidic bonds are seen in between adjacent sugars in a polysaccharide or carbohydrate chain. Next one, amino acids that cannot be synthesized within our body but can be obtained through diet are called. There are two types of amino acids, essential amino acids and non-essential amino acid. Non-essential amino acid can be prepared, can be synthesized within our body. Our body can prepare this. So, essential amino acids, since our body is unable to produce, cannot be produced. That must be supplied through the diet. So the amino acid that must be essentially present in the diet are called essential amino acid. Amino acid that cannot be synthesized are called essential amino acid. Non-essential amino acid can be synthesized. Next one, which among the following is not a polymer of glucose? So cellulose is a polymer of glucose. Glycogen is a polymer of glucose. Starch is also a polymer of glucose. But inulin is the answer. Inulin is a polymer of fructose. It's a polymer of fructose. Next one regarding the sugar present in DNA. In DNA, the sugar is called deoxyribose. What is ribose? Ribose means ribose sugar is C5H10O5. In deoxyribose, one oxygen is detached. That means C5H10O4. This is detached from the second carbon atom. So one oxygen is detached from the second carbon atom of the ribose so that it becomes deoxyribose. That kind of a sugar is present in DNA. Hemoglobin molecule shows dash structure of a protein complex. The linear arrangement of amino acids is called primary structure. The alpha helix or the beta pleated sheet, this is called secondary structure. The irregular arrangement is called tertiary. The combination of many tertiary is called quaternary. So hemoglobin actually comprises of 2 alpha chains plus 2 beta chains. So it is a quaternary protein complex. Then a topic from enzymes. When we consider enzyme substrate complex and enzyme product complex, which of the following statement is correct? What are enzyme substrate and enzyme product complex? Enzyme molecule first combine the substrate molecule forming a complex. That complex is called enzyme substrate complex. Enzyme substrate complex get converted into another complex. That complex is called enzyme product complex. Finally, the enzyme product complex dissociate, releasing enzymes and products. This is the nature of enzyme action. So two complexes are produced, two intermediate complexes are produced. One is called ES complex, another one is called EP complex. So ES complex is a temporary unstable complex, so that it get converted into EP complex. So this is unstable. This is unstable. The EP complex produced is also unstable, that is why it get converted into enzymes and product. So the EP complex produced is also unstable. So here both ES and EP are unstable. That means ES complex, enzyme substrate complex, as well as the enzyme product complex, both are unstable complexes. Next one regarding competitive inhibition. In competitive inhibition, dash and dash will be structurally similar. What is competitive inhibition? Suppose this is enzyme. This is the shape of the enzyme molecule. This is the shape of the substrate molecule, and this is the shape of the inhibitor molecule. Think like that. If there is a combination between enzyme and substrate occurs, that leads to ES complex and finally forming the products. ES complex, then EP complex, product formation. If there is a combination between enzyme and inhibitor, the complex produced is called enzyme inhibitor complex, that cannot be converted into product. That means the enzyme inhibitor complex produced is stable. Okay, ES complex is unstable, we already studied. It is unstable, that means it converted into product. So here substrate can attach with the active site of the enzyme, inhibitor can also attach with the active site of the enzyme, only because there is a structural similarity between the enzyme molecule and the inhibitor molecule. So actually there will be a competition between substrate and inhibitor to attach with the active site of the enzyme. If the substrate wins in the competition, product will be produced. If the inhibitor wins in the competition, the product formation is inhibited or inhibition will be there. So this kind of inhibition is called competitive inhibition. So in competitive inhibition, the structural similarity is between substrate and inhibitor. So the answer will be substrate and inhibitor. The next one regarding the type of category of enzyme. The category of enzyme used in the reaction Sg plus S dash gives S plus S dash G. Here S having a side group called G combines with the S dash. So this is one substrate, this is another substrate. What are the products produced? Product produced will be S plus S dash G. That means the G group attached to the first substrate called S is now present attached with the second substrate called SG, S dash. That means there is a transfer of group from one substrate to another substrate. So this enzyme is called transferase. The functional group transfer is possible here. So that enzyme is called what? Transferase. Next one, the organic compounds that are tightly attached to apo enzyme are called. So there are two types of enzyme. In one enzyme, only protein part is there. That kind of enzyme is called simple enzyme. The simple enzyme comprises of only protein part. In some other enzyme, a protein part will be there. In addition to protein part, a non-protein part also is there. So that enzyme is called conjugated enzyme. That enzyme is called conjugated enzyme. So in the conjugated enzyme, the protein part is called apoenzyme. The non-protein part is called cofactor. Okay, cofactor is the non-protein part of a conjugated enzyme. And there can be three different types of cofactors. the organic part which is tightly attached. Organic part which is tightly attached is called a prosthetic group. Organic part which is loosely attached is called a coenzyme. So then some metal may be used as activators. So here the correct answer, organic compound which are tightly attached to the apoenzyme. Apoenzyme is the protein part. Is called prosthetic group. Next one regarding the metallic activator of a carboxypeptidase enzyme. Carboxypeptidase enzyme is activated by zinc. Next question, RNA differs from DNA. That already discussed. In RNA, there will be adenine, guanine, cytosine, uracil. In DNA, the nitrogenous present will be adenine, guanine, cytosine, thymine. So RNA differs from DNA in having uracil instead of thymine. In having uracil instead of thymine. Not thymine instead of uracil. Okay. Next, regarding the KM value. What is KM value? KM value means the Michaelis-Menten constant. That means the influence of substrate concentration in enzyme action. Okay. So here the velocity of reaction, here the substrate concentration. So this is a maximum velocity. Maximum velocity is called Vmax. So this will be the half Vmax. Then this will be half Vmax. The substrate concentration at which the velocity of a reaction attains half of its maximum value is called a Km value. The substrate concentration, when we are increasing the substrate concentration, the velocity initially increases and reaching a maximum point of activity, maximum point of velocity at a particular substrate concentration. Then even though we are increasing the substrate, the activity will be in the same pace. So the substrate concentration at which the velocity of a reaction attains half of its maximum value is considered as what? Km value. So Km value Vmax by 2, half Vmax. The last question. Two statements are there. Statement 1, enzyme reduce the activation energy of a reaction. Activation energy means the minimum energy at which the reaction is possible. When we are adding enzymes, the activation, those reactions which require higher level of activation energy can be conducted inside the body at normal temperature. So enzymes always reduce the activation energy of a reaction. That's a right statement. Then the most abundant enzyme in the biosphere is Rubisco. In your textbook it is written that the most abundant protein in the biosphere is Rubisco. But Rubisco is an enzyme in photosynthesis, in C3 cycle. It is an enzymatic protein. It is the most abundant enzyme. So both the statements are right. Enzymes reduce the activation energy of a reaction. Rubisco is the most abundant protein or most abundant enzyme in the entire world. So both statement 1 and 2 are true. C. So by this we have discussed around 20 questions from the chapter Biomolecule. The chapter is very important. So you have to go through all the points seriously. Okay, thank you.